Hello friends, welcome on my YouTube channel Actual Biology. Today we will discuss about the GPCR-plc pathway, means GPCR protein, G protein coupled receptor, and PLC means phospholipid C. Okay, mostly it will work into the means muscles contractions. Okay, sperm moves towards the ova. Okay, so these are the some examples of GPCR-plc pathway. Okay. So we are going to discuss about the what is the role of receptor and receptor molecules. After binding the receptor molecules, how the receptors work. Okay, so this is the suppose that this is the plasma membrane. Okay, this is the plasma membrane of a cell. Okay, so before this I completed GPCR pathway, G protein coupled receptor pathway. You can check it into the description box. Okay, so this is the plasma membrane. In plasma membrane, one protein present, means one protein is 7 transmembrane protein present. Okay, in 7 it is a single protein. It is 7 transmembrane protein in which 3 external loops and 3 internal loops. Okay, so 1, 2, 3, 4, 5, 6, 7. These are the 7th transmembrane protein. Means it is a single protein. Okay, so this is protein, and internal loop 3 has GEF activity. It is very important work of internal loop 3. Okay, GEF means G nucleotide exchange factor, and what is the role of GEF we are going to discuss. Okay, so the another protein is embedded into the plasma membrane. What is this protein? This protein is G protein. Okay, G protein has 3 subunits: G alpha, G beta, and G gamma. Okay, these proteins are embedded into the plasma membrane in the heterotrimeric form. Okay, heterotrimeric form means 3 proteins are in different form. Okay, so G alpha, G beta, and G gamma, these are in a complex form. Okay, then here it means suppose that here is the receptor, because in all the transmembrane proteins, or we can say in the over the plasma membrane of cell. Okay, all the means receptors are present. Okay, so means here the receptor molecules bind, and after the binding of receptor molecules, cell will respond. How it responds? Okay, so this all we are going to discuss. Okay, suppose that here the receptor molecules bind at the receptor, because at the proteins, we can say at the surface of the protein, receptors are present. The protein, which is the protein 7 transmembrane protein, it is a single protein. Okay, so after the binding of receptor molecules, G means internal loop 3 activated, and what is the role of internal loop 3? GEF means G nucleotide exchange factor. Normally at the G protein, G alpha has binding GDP. Okay, when it is into the heterotrimeric form, normally GDP binding at the site of G alpha protein. Okay, when receptor molecules bind at the receptor, internal loop 3 has activated. Then after the activation of internal loop 3, binding means G nucleotide exchange factor. What is the role of G nucleotide exchange factor? It will exchange the GDP into the GTP. Okay, here it will exchange. It is a very important point. It will exchange, not convert. Okay, it will exchange. After the binding of GTP, here G alpha gets separated from the G beta and G gamma. Okay, so this is the, it will separation takes place. Okay, so after the separation, G alpha protein activate the another protein. It is a PLC. which is embedded into the plasma membrane. Okay, so what is PLC? Phospholipid C proteins embedded into the plasma membrane. And phospholipid C has 4 classes. One is phospholipid C beta, phospholipid C gamma, phospholipid C delta, phospholipid C epsilon. Okay, so what is the role of PLC? It is a very important role into this this pathway. Okay, so PLC after the activation of G protein, G alpha protein activate the PLC. Means G alpha protein activate the PLC means PLC beta class of the PLC get converted PIP2 into the DAG and IP3. It is a very important step of this pathway. Okay, so this is the PLC. Okay, means PLC beta. What is the work of PLC beta? It will convert PIP2 into the IP3 and DAG. What is DAG? Diacyl glycerol. And what is IP3? IP3 is inositol triphosphate. Triphosphate. Okay, so and what is PIP2? PIP2 means phosphatidyl inositol diphosphate. Okay, diphosphate in means 4,5 diphosphate. In triphosphate 1,4,5 triphosphate. Inositol 1,4,5 triphosphate. Okay, so what is the work? What will be the work of the IP3 and DAG? Okay, so this is a very important point here. This step is occur into the plasma membrane because there is no place, so I written it here. Okay, so what is IP3? These steps are occur into the plasma membrane. Okay, plasma membrane means PIP2 occur in plasma membrane, PLC occur in plasma membrane. So PLC beta get converted PIP2 into the IP3 and DAG. Okay, after the conversion of IP3 gets released into the cytoplasm. Okay, then IP3 get released into the cytoplasm. Okay, into the cytoplasm. And what will be the work? We are going to see here. Okay, so here DAG, diacyl glycerol, remains embedded into the plasma membrane. Here DAG convert means DAG remains into the plasma membrane. And what will be the work of DAG? First we are going to check the role of IP3. Okay, so what will be the work of IP3? IP3 goes to the endoplasmic reticulum because this is the part of cytoplasm. Okay, cytoplasm of the cell. Okay, so IP3, what will be the role of IP3? IP3 getted calcium release channel. It will open the IP3 getted calcium release channel, IP3. Okay, because IP3 is work as a ligand here. Okay, because here it is channel, IP3 gets bind to this channel and it will open the IP3 calcium release channel. After the binding of IP3, calcium move out from the IP3. Okay, calcium release into the cytoplasm. And after the release of calcium, what will happen? Okay, so here DAG remain still into the plasma membrane. Okay, so into the presence of calcium, DAG gets activate the protein kinase C. Okay, in the absence of calcium, DAG will not get converted into the protein kinase C. Okay, after the release of calcium, DAG get activate. We can say DAG get activate and it activate the protein kinase C. Protein kinase C means in the presence of all this work happened into the presence of calcium. Okay, so calcium concentration in the cytoplasm will increase. Okay, calcium concentration will increase. So what will happen? So after the activation of protein kinase C, kinase means it will phosphorylate the particular protein. Okay, after the phosphorylation of protein, protein gets activated and respond into the cell. Okay, so this is the because after the phosphorylation, we all know, after the phosphorylation of proteins get activated or inactivated. If some proteins are in activated form, after the phosphorylation the proteins will get activated form. Okay, protein gets activated and respond into the cell. Okay, so this is the overall process of GPCR PLC pathway. I hope this video is helpful for all, and if you like the video, please hit the like button. Thank you so much.